first part we studied about the structure of the male reproductive organ, that is the androgen, and the structure of the microsporangium, etc. Okay, now we are going to discuss about what is microsporogenesis, that is how the microspores produced, and the structure of the pollen grain and how the pollen shed, etc. Okay, microsporogenesis. Okay, what is microsporogenesis? As the name said, the microsporogenesis, that is the process of formation of microspores. The process of formation of microspores from the microspore mother cell through meiosis is called the microsporogenesis. That is the process of formation of microspores from the microspore mother cell through meiosis is called the microsporogenesis. Okay, in the last class, that is in the first part, we studied about the sporogenous tissue. We studied about the sporogenous tissue, that is inside the inside the microsporangium, the homogeneous cells are present. These homogeneous cells are called sporogenous tissue. This sporogenous tissue, they are differentiated into form microspore mother cell, microspore mother cell, or pollen mother cell, This microspore mother cell or pollen mother cell, they differentiate, they undergo meiosis and form 4 haploid microspore tetrad. Okay, the sporogenous tissue of the microsporangium, they are differentiated into form microspore mother cell or pollen mother cell. This pollen mother cell, they undergo meiosis and form haploid microspore tetrad. This haploid microspore tetrad, on dehydration, they dissociated to form 4 microspores, 4 haploid microspores, and these 4 microspores, they become 4 pollen grains. That is, each microspore developed into form pollen grain. That's all about the microsporogenesis. That is, what is microsporogenesis? Microsporogenesis is the process of formation of microspores from the microspore mother cell through the process of meiosis is called the microsporogenesis. That is the sporogenous tissue. First of all, the sporogenous tissue, it is differentiated into form microspore mother cell or pollen mother cell, and this microspore mother cell, they undergo meiosis and form haploid microspore tetrad. This haploid microspore tetrad, on dehydration, they form 4 microspore, and each microspore then developed into form pollen grain. Okay, that's all about the microsporogenesis. Okay, each microspore developed into form a pollen grain. Okay, each microspore developed into form a pollen grain. Now we are going to discuss about detailed structure of a pollen grain. Okay, what about pollen grain? The pollen grain, which represent the male gametophyte, pollen grain which represent the male gametophyte. Each pollen grain consisting of 25 to 50 micrometer in diameter. Each pollen grain consisting of 25 to 50 micrometer in diameter. And what is the shape of pollen grain? Most of the cases spherical in shape. That is, the pollen grain, it represents the male gametophyte. It is spherical in shape and it is measuring about 25 to 50 micrometer in diameter. In most of the cases, the pollen grain is consisting of two wall layers. Which are they? The outermost layer is called exine and the inner layer is called intine. Okay, the outermost layer is called exine and the inner wall layer is called exine and intine. Okay. Okay, what is the speciality of the exine? Okay, the exine, it is made up of sporopollen. What is the speciality of the sporopollen? It is one of the most resistant organic material. It is one of the most resistant organic material. It can withstand high temperature and can withstand strong acids, alkali, and no enzyme can degrade this. No enzyme can degrade this material. That's all the specialty of the exine. What are the specialties? The exine, it is made up of sporopollen. The sporopollen, it is one of the most resistant organic material that it can withstand high temperature and it can withstand strong acids, strong alkali, and no enzymes can degrade this material. Okay. In the case of exine, there is a small aperture is present. In the case of exine, there is a small aperture is present, but the sporopollen is absent. This aperture is called germ pore. Through this germ pore, the pollen tube develops. Through this germ pore, the pollen tube develops. Okay, what is the function of pollen tube? The pollen tube helps in the release of the pollen grains during fertilization. They helps in the release of the male gametes during fertilization. That is, through this pollen tube, the male gametes goes to the outer side during the fertilization. Okay, that's all about the exine. The exine, it is made up of sporopollen. It is one of the most resistant organic material. It can withstand high temperature. It can withstand strong acids and alkali, and no enzyme can degrade this material. In the exine, there is a small aperture is present. This aperture is called germ pore. Okay, this germ pore helps in the formation of the pollen tube. The pollen tube helps in the release of the male gametes during the time of fertilization. Okay, that's all about the exine. Okay, exine is one of the resistant material. It is sporopollen is present in the exine. Okay, then what's about the intake? Intake, this very thin and continuous layer, and it is made up of cellulose and pectin. Intake, it is very thin layer and it is very continuous layer, and it is made up of cellulose and pectin. That's all about the two wall layers, exine and intake. Okay. When the pollen grain develops, pollen grain develops, it become two cell stage. Okay, it become two cell stage. The upper cell is called vegetative cell and lower cell is called generative cell. The vegetative cell, it have dense cytoplasm and it have abundant food reserve. And it have large and irregularly shaped nucleus. Okay, in the case of generative cell, it is very small. It floats in the cytoplasm of the vegetative cells. Okay, when the pollen grain become mature, it will become two cell stage. That is the uppermost cell is called generative cell and the lowermost cell is called vegetative cell. The vegetative cell, it have dense cytoplasm. That is, it have abundant food reserve and it also have a large and irregular shaped nucleus. In the case of generative cell, it is very small in size. It floats in the cytoplasm of the vegetative cell. Okay, in most of the cases, that is in the case of 60% of the angiosperms, they shed at this, the pollen grain shed at this two cell stage. That is, in the case of angiosperm, 60% of the angiosperm, they shed at this two cell stage. That is, then the remaining 40% of the angiosperm, they shed at the three cell stage. That is the generative cell. This generative cell, they mitotically divided and form two male gametes. They mitotically divide to form two male gametes. There is altogether three cells. 1, 2, 3. Okay. That is, in most of the cases, that is in 60% of the cases, the pollen grains, they shed at the two cell stage. Remaining, in the case of remaining plants, they shed at the pollen grains shed at the three cell stage. That is, this generative cell again divided mitotically and form two male gametes. That is altogether three cell stage. Okay, that's all about the structure of the pollen grain. Okay, thank you. That is all about the structure of the male reproductive organ. This is structure. We studied about the structure of the androecium. That is the structure of stamen. Then the structure of the microsporangium. And what is how the microspores are formed, and how the microspore become pollen grains, and how the pollen grain shed from the plant. Okay, that's all about the male reproductive structure. Okay, thank you.